The final part of chapter 15 is pretty straightforward. Although it takes a lot of pages to describe what's happening, the English language used is straightforward, with a few scientific words sprinkled in here and there to clarify what's going on. So secretory pathways simply follows the passage of proteins, lipids, and other molecules through a cell's migration system. Students should be aware that the secretory pathway begins at the endoplasmic reticulum and migrates through the Golgi, through vesicles, to the plasma membrane, where the proteins are secreted. During the passage of these proteins, at the early stages, some common events take place, such as illustrated in this slide. The addition of oligosaccharides, sugar molecules, to the polypeptide chain takes place in the lumen of the endoplasmic reticulum. So this event is unique to the lumen. Very rarely do sugar molecules become added to polypeptides on the cytosolic phase. And when they do, they normally involve individual residues, not chains of residues as indicated here. In certain circumstances, the structure of the finished polypeptide, the protein, can be stabilized by the formation of disulfide bonds, which involves neighboring cysteine amino acid side chains using their sulfur atoms to form disulfide covalent bonds. These are placed in proteins that require additional stability, normally those that are eventually secreted from the cell. Please always keep in mind that there's a lot of quality control taking place at each stage in the secretory pathway. And exit from the endoplasmic reticulum is one of those very important quality control steps. Within the lumen of the endoplasmic reticulum, there are present many chaperone proteins whose task is to bind to any misfolded polypeptides and to retain them in an environment that encourages proper folding, and should that fail, the protein is then destroyed. Only proteins that are properly folded are allowed to progress on their journey to be secreted from cells. Additionally, there are other quality control steps on top of the chaperone system. Should the chaperone system become overwhelmed with misformed proteins, then the ER has the capacity, a more profound response known as the unfolded protein response. This leads to the generation of further endoplasmic reticulum by growing in size, as well as the generation of more chaperone proteins and also the production of other proteins designed to clear the lumen of the endoplasmic reticulum. In some cases, as this diagram shows, the system can also produce proteins which inhibit further protein synthesis. So the response is quite paramount. If you draw your attention to the membrane here in gray, you will see that these proteins which detect the formation of unfolded proteins in the lumen of the ER are in fact transmembrane proteins which can communicate their findings across the membrane into the cytosol, and that can lead to activation of genes and other biochemical events that are detected within the endoplasmic reticulum. Those proteins which acquire the correct folding are then passaged on to the Golgi apparatus. The students need to know the Golgi apparatus in some detail here. And as this figure shows, the Golgi apparatus can be divided into regions, and those regions are depicted here. So the side that's facing the endoplasmic reticulum, and indeed the nucleus on this side, would be called the cis Golgi network. This receives vesicles from the endoplasmic reticulum. The middle part is called the medial cisternae, and a cisternae is simply a sac is simply a sac of the Golgi apparatus. And on the other side, facing towards the plasma membrane, we have the trans-Golgi. So new material enters on this side, and as it progresses through by being budded off and exchanged between cisternae, modifications are continually taking place so that mature vesicles end up being generated on the trans-Golgi network. Students should be aware that any proteins which still carry ER retention signals can be returned from the Golgi back to the endoplasmic reticulum in those vesicles that are headed in that direction. So only mature non-ER retention proteins are allowed to transverse through the Golgi apparatus. And as the material passes from different areas of the Golgi to new areas of the Golgi, unique modifications are taking place, especially to the sugar molecules that we spoke about earlier when it came to the lumen of the ER. So by the time you get to these vesicles in the trans-Golgi network, those on the right-hand side may be headed towards the plasma membrane, those in the middle may be headed towards endosomes, whereas those on the left-hand side may be headed to just storage vesicles within the cytosol. Each one with this unique set of proteins and lipids. At this point, it's important for students to focus on the How We Know pages, those two pages in the gray color. By reading over these pages, students should get an appreciation of how in the past we have performed experimentation to get to the understanding that we have today. This figure, taken from there, shows us that signal sequences can be radioactively applied to material that allows us to then follow the location in which the proteins end up doing their processing. So the radioactive material becomes a visual marker of where the protein is directed. The findings from such experiments have increased our understanding to the point that we now know that under normal circumstances, the process of transfer from the endoplasmic reticulum via the Golgi to the membrane takes place in a straightforward, simple pathway. But in some cells, which have blocks at various points, we do find that proteins begin to accumulate in the ER. Those proteins do not get to the endoplasmic reticulum. And those cells then become very valuable in understanding why the proteins are held back at that location. Similarly, other mutations show us that the block is further along and that the secretory vesicles do not end up near the plasma membrane. So those cells can be investigated to deduce why that process is holding onto the protein in the Golgi apparatus. And then finally, there are even mutations where the vesicles do appear in the cytosol, but they for some reason do not blend with the plasma membrane. So results from these type of mutant experiments are very valuable in deducing how proteins migrate from secretory systems. This figure here is a great example of a time-lapse photograph sequence showing how in a yeast cell proteins accumulate in different places over different periods of time and that they follow this pathway. So here's the endoplasmic reticulum, and here are those proteins leaving the endoplasmic reticulum in vesicles. Those vesicles head to the Golgi, and from the Golgi they are then passed through vesicles to the plasma membrane. Students should be familiar with the word exocytosis, which is a process by which material from within the cell is released to the exterior environment. So focusing on that part of the cell, we can see that there are two general pathways, and they are mentioned here. There's a standard pathway that operates in all cells, and that's called the Constitutive secretion pathway. And all cells perform this particular route. Here, vesicles pinch off the Golgi, they travel along the cytoskeleton, and as they do, the contents of the vesicle may mature and change, so that when final delivery is made, the plasma membrane of the vesicle is added to the plasma membrane of the cell, and that would include the black dots, which are proteins. So this is one way of delivering new proteins to the plasma membrane. Anything in the lumen of the vesicles is then fair game to be released either into the immediate environment around the cell matrix, or beyond. The alternative pathway, which is called the regulatory secretory pathway, is employed by those cells that have a task of releasing secretory proteins in large dosage. So this is how insulin would be secreted by pancreatic cells. So during this pathway, the vesicles actually are held up near the surface of the plasma membrane and only fuse with the plasma membrane when a signal is received either from the outside or from the inside of the cell. This pathway is very beneficial in that during the formation of these vesicles, their contents can be highly concentrated, and the vesicles can be parked here just under the plasma membrane for periods of time until a signal is thus received. Let's now turn our attention to endocytic pathways. Endocytic pathways bring material into the cell. Now, there's a number of mechanisms that operate, but there are two main ones: phagocytosis and pinocytosis. So phagocytosis is the engulfing of large material from the surroundings of the cell and bringing that material into the cell. So classic examples of this are white blood cells consuming bacteria. So here we have a macrophage, which is a white blood cell, and it's engulfing this dividing bacterium that is found in the bloodstream. So what happens is that there are receptors on the surface of the white blood cell. Once those receptors make contact with the bacterium, they then send out these finger-like extrusions of the plasma membrane called pseudopods, and the pseudopods will link together on the far side, therefore encompassing the bacteria in some type of endosome. And then those endosomes are then imported deeper into the cytosol to be then digested, and the bacteria are killed. The panel B shows us a white blood cell, macrophage in fact, consuming two dead red blood cells. So as it says in the book, every day lots and lots of red blood cells are ending their lives by being phagocytosed. And once this material enters the cell, it then is digested down into amino acids, nucleotides, carbohydrates, and lipids, the building blocks of new things within the cell. At a smaller scale, fluid and small molecules are taken up by pinocytosis, and most cells are performing pinocytosis constantly by sipping their extracellular fluid and bringing that into the cell by means of the endocytic pathway. An alternative mechanism is receptor-mediated endocytosis, which provides a specific route into animal cells. And that is indicated in our case by the uptake of cholesterol. Cholesterol is indicated here as the red molecule. So the red molecule cannot travel in a liquid environment, an aqueous environment, without some type of protection. And you can see here the green structure is a protein shell that allows the cholesterol to travel through the blood. Upon delivery to cells, there are receptors, very much like the middle part of this chapter, where we had the cargo receptors. Well, these are an example of those cargo receptors. So the cargo in this case is LDL. That stands for low density lipoprotein. And everybody knows about this because when you go to the doctor to have your blood tested, they're testing for the presence of these materials in your blood. Regardless, let's get back to this. So the cargo receptors are, in this case, LDL receptors, and those LDL receptors will bind to the proteins and then start concentrating in a region of the membrane known as a raft or domain. And that's where you get the adaptins and the clathrin molecules forming, and they form a vesicle. And the vesicle is budded off by proteins such as dynamin. And the vesicle travels along the cytoskeleton pathway, and as it goes in, it may even fuse with an endosome or become an endosome itself. And endosomes exist in an early phase and a late phase. So these early phase endosomes will then allow the cargo receptors to be recycled back to the surface of the cell. So this flow continues so that there's a constant uptake of LDL. The cargo itself is then directed down a pathway where it becomes a late endosome, and then finally it matures into a lysosome, or it fuses with an existing lysosome. Either way, the end result is that as the content of the lysosome begins to take action, the protein shell is digested away, releasing within it the cholesterol molecules, which are then small enough to be absorbed through protein transporters that are embedded in the membrane of the lysosome. And then for the first time, those chemical molecules are found in the cytosol of the cell, and then they can be used to drive other biochemical processes that the cell needs. This is a particularly important diagram because it illustrates alternative pathways that can be taken by components of the endocytic pathway. So as we saw earlier with the Ldl receptors, there's a recycling event where membrane can be recycled quickly back to the plasma membrane. There's an alternative pathway that leads to degradation ending in a lysosome. So the lysosome is the end of the road. But there's also a third pathway that you should be familiar with. And this pathway can take these vesicles and deliver them to an alternative destination within the same cell. In this case, you may be familiar with this figure from another chapter. Here we have a cell lining our digestive system. So the food will be moving at the top here, and the blood supply will be down at the bottom of the slide. So these lining cells of our gut, they have tight junctions which prevent the passage of molecule between the cells, between neighboring cells. So everything that has to pass from one side to the other has to pass through the cell, and that's called transcytosis or transcellular transport. So in this case, vitamins could be picked up from the gut surface and then delivered to the basolateral plasma membrane, from where they can easily diffuse into the blood supply to be delivered to other cells. And then lastly, we have to discuss something about lysosomes. And the lysosomes are the cell's principal sites of digestion, just like mini stomachs that the cells have. So any material that needs to be digested is primarily directed to lysosomes. And lysosomes contain enzymes for degrading all types of molecules that are commonly found in food particles or structures within the same cell. The important thing to realize is that lysosomes generally have a pH that's pretty acidic, and they maintain that by doing something that we're familiar with from previous chapters, in that they pump protons using a proton pump into the lumen of the lysosome. So there's a proton pump here, and that is a primary proton pump because it uses ATP energy in the cytosol directly to power the movement of protons. Now, if you're familiar with the word ASE, that means these are enzymes. So all these things are enzymes, and the prefix tells you what it digests. The final figure here shows us that material that's endocytosed ends up in the lysosomes, becoming broken down into such small particles that the cell can then import them across a membrane into the cytosol to join its other chemistry. An alternative, as we saw earlier, is phagocytosis, where large substances are imported into phagosomes, and the phagosomes merge together with endosomes and then eventually end up in the lysosomes. And the lysosomes can actually merge with phagosomes and endosomes directly, as indicated by the green arrows here. And finally, the third pathway is old material that the cell no longer needs. For instance, this mitochondrion is no longer functioning properly, and the cell feels that it can be degraded. So membrane will form around the mitochondrion and lead to the production of a vesicle, and that's called autophagosomes or autophagosomy. And once the organelle has been surrounded, it is then going to be fused with a lysosome, and the content of the lysosome will digest the content of the autophagosome.